So today we're gonna focus down on chapter 7 of my new book, The Quest for New Knowledge. The title of this chapter is the genetics of cellular proliferation. As you may recall, that's really what I wanted to find out about when I first set out in the early days of my scientific career in the early to mid 70s. I wanted to know what was regulating cellular proliferation, and ultimately to get to cancer, to understand what was wrong with cancer cells. But in order to do that, I realized that I needed to study normal cell regulation of normal cell growth in order to understand what would happen when it became abnormal. And this is sort of a segue from chapters 5 and 6. It all takes place in the 1980s. And so chapter 5 was all about the interleukin-2 / receptor structure activity relationships in terms of trying to figure out how the ligand bound to the receptor. We were really focused down on the structural biology of that. And simultaneously, we got into these immunotherapy experiments as a consequence of my, what do I want to say? I was very dismayed by Steve Rosenberg's use of IL-2 in the clinic because I thought he did it all wrong. He didn't know what he was doing, really. He didn't treat it like a hormone like it should have been. He treated it like a toxic chemotherapeutic agent, which it is not. Although what he did by giving very, very high doses of it, he made it a toxic agent. And so while all these things were going on in the lab and in collaboration with Jerry Ritz and Mike Caligiuri and so forth down at Boston, in the early 80s, a phenomenon was reported that really pinned down for the very first time that cancer was caused by mutations in genes. Up until that time, the favorite hypothesis was that viruses cause cancer. And of course, viruses can cause cancer, but they're not the common cause of cancer. We know as a consequence of what I'm going to tell you about today. Now we know that there are genes inside cells that if they happen to get a mutation in those genes, you don't need a virus. That mutation can cause the cell to undergo what the scientists called a malignant transformation. So you transform from a normal cell into a malignant cell. And so in the early 1980s, Doreen Cantrell and myself and Martin Gullberg, who was another postdoc from Sweden, who was very good, we found ourselves at the Hanover Inn again. That seemed to be our resting place because we were entertaining Frank Rossetti. He was up from Bethesda for a couple of weeks, and we were doing experiments together on the IL-2 receptor. And so we sort of decamped to the Hanover Inn one evening and sat there drinking beer, and we got into a conversation about these oncogenes, which were just being splashed all over the place in the scientific literature anyway. The story about these genes is interesting. It really starts and as a consequence of the discussions we had that evening, I sort of wondered whether or not we could use the IL-2 T-cell model cell growth system that Doreen had developed in order to get at what was going on inside the cells to move them from a resting stage to a proliferative stage. And because cancer cells proliferate sort of so-called willy-nilly or abnormally, that it seemed that if people had discovered genes that were abnormal in cancer cells, then maybe this is a time for us to bring these two things together: normal cell growth and malignant cell growth. And the story about the oncogenes goes back to 1964. There was a fellow by the name of Howard Temin, who was at the University of Wisconsin in Madison, and he came up with a new hypothesis. He said that he was studying sarcomas in chickens. It was caused by actually the very first retrovirus that had been discovered, Rouse sarcoma virus, and this had been discovered by Peyton Rouse in 1910. And so we'd known about it for over half century or more already. And if you had this Rouse sarcoma virus, RSV, inoculated it into chickens, and particularly newborn chickens, they would ultimately get cancers. They would get big solid tumor sarcomas. Temin came up with a hypothesis to explain viral carcinogenesis, how viruses could cause cancer. And he said, and his hypothesis was that, well, the viruses had genetic material in their total gene sequences and so forth that they may have been picked up along the lines and of infecting many people or many chickens and so forth that ultimately were abnormal and that could cause the sarcomas. Then as time, and then he got really ridiculed for this. I mean, because people said, okay, fine, Howard. And Howard was a scientist. He was a typical nerdy scientist. And he was a serious scientist, and everybody knew that, and he was very well respected. But boy, he took a lot of heat at national meetings and international meetings, and the field of, I guess, the viral types primarily, because that's who he was talking to. He didn't like this hypothesis because his hypothesis was that these tumor viruses, like Rao sarcoma virus, were made, their genetic material was made out of RNA. It wasn't made out of DNA, which is what our genetic material is made out of. And the central dogma in molecular biology at the time, as basically coined by, I think, Francis Crick, the central dogma was that information flowed only from the genes in DNA to RNA that took the information from the genes and brought it into the cytoplasm. And then it transcribed this information, transcribed from the nucleus, and then translated it into protein in the cytoplasm, which then either made up parts of the cell or excreted into the environment and so forth. So that was the central dogma. It went from DNA to RNA to protein. Well, that was just a little bit of a problem for Temin's hypothesis, because then how could an RNA virus get into the DNA? And he hypothesized that there was an enzyme that had never been described before that ultimately became called reverse transcriptase, meaning that it took the information from RNA and it copied it into DNA and then got inserted into the DNA of the cell. And then people laughed at him, said he was crazy. And this went on for 4 or 5 years, and it wasn't until 1971 when he not enlarged, but he improved his hypothesis. And he said, not only did viruses have the abnormal genes in them that were causing cancer, and they got reverse transcribed. And incidentally, he ultimately went on to get the Nobel Prize for that whole thing, along with David Baltimore. And that's a different story that we won't get into right now. But in 1971, he said, well, what's really going on with all these cancers and so forth is the virus comes inside the cell. And there has to be genes already in the cell that could potentially cause cancer. And so he called this his cellular proto-oncogene hypothesis. And that was that you can forget about the viruses. There are genes in the cell that have the potential to transform the cell to be malignant that he called proto-oncogenes. Well, that was 71. In 76, there was a group out in California that were dealing with Rao sarcoma virus and so forth. And the names of these people there were two of them, Mike Bishop and Harold Varmus. And they essentially proved Temin's hypothesis. Well, and so all the people knew about this and so forth. But it wasn't until 1979-80, and this was what's precipitated our conversation in the bar at the Hanover Inn. There were 2 reports that came out from 2 different investigators, Howard Weinberg at MIT, and Jeffrey Cooper. And I think that he was at BU, so the same city, but different people. And they basically they devised an assay for this malignant transformation effect, and it was a visual assay. What they did was that they took DNA from cancer cells, ground them all up in a gamish and so forth, and separated out the DNA molecules and put them into cell cultures of mouse fibroblasts. and then would wait a few days or a week or so, and then the cells would transform. So this is why it's ultimately called the transformation assay, and they would no longer look the same. They would start to be funny looking, and their growth characteristics change such that they piled up on top of one another and just kept on growing and growing. And they would form foci. So if you diluted out the DNA far enough, you could get foci of these transformed cells, and you could count them, so that therefore they had a quantitative assay in this whole thing. Well, that just created a huge explosion of people interested in this whole thing, because they had this new cellular transformation assay, which was a visual assay, really. Just looking down the microscope and looking for these, counting these foci. So a year or so after that, a new postdoc came into the lab, Julie Stern, and she was a young woman who had gotten her PhD at Dartmouth in a molecular biochemistry lab, studying molecular trafficking inside cells and so forth. And so she came into the lab, and we told her all about this interleukin-2 stuff and the T cells and that sort of thing. And so she took upon herself to get involved to see whether or not we could use the Doreen's IL-2 T cell growth system to look for some of the expression of these oncogenes. Because by this time, a year or two went by, and by this time there may be, 20 different genes that had been isolated and shown to work in this cell transformation assay to cause cancer. At least that's what they thought they were looking at. So she started off and she screened about a dozen, I think, of these new oncogenes in the T cell system. And so the nice thing about the T cell system is that you could grow these T cells that had IL-2 receptors in IL-2, and you can grow enough of them so that you had enough cells to work with. And then you could wash them out of IL-2 and put them in fresh medium. And when you did that, they would just wind back down, and they would all they change morphologically from big blastoid cells down to small resting lymphocytes again. But then they wouldn't die. I mean, they would die eventually if you left them out of IL-2 for very long. But then you could give them IL-2 back again, and then they would synchronously, meaning almost all the cells in the culture would start to replicate their DNA and undergo mitosis almost simultaneously. So you had a very nice system where you could do kinetics or timed. You could stop the reaction after 1 hour and 2 hours and so forth and so on. And you could look for the appearance of these oncogenes that had been reported. And to make a long story very short, she found several of these oncogenes that were expressed in a sequential manner before the cells went into DNA replication and then subsequently into mitosis. So basically, she proved that our hypothesis that you could use the T cell IL-2 system to study the genes that were involved or were thought to be involved in activating the cells to grow. Her experiments focused down on one particular oncogene. We found it had been reported, and also we found the same thing, that there were three of these oncogenes that were expressed after you activated the T cell receptor, but they were also expressed after you activated the IL-2 receptor. And I didn't particularly like that whole story because of the fact that I knew that structurally the T cell antigen receptor and the IL-2 receptor were different. And so I expected them to activate a different set of genes. And ultimately I was right about that. But there were some genes that were activated by both receptors. And so that was what. But Julie found that there was one particular gene, and it was called MYB, M-Y-B. The people used acronyms to name these genes. This was named after myeloblastosis virus, which was a different RNA tumor virus that caused leukemia in chickens. It's interesting how biologists, as long as it's alive, you're going to work on it. So you extrapolate from, chickens are basically the descendants of dinosaurs. And so you might think that they were a little bit far removed from human beings. But anyway, so she worked on MIB, and she found that MIB was only activated to be expressed by IL-2, and the IL-2 receptor was not activated by the T-cell receptor. So about that time, I had been invited to go out to UCSF, University of California at San Francisco, to give a seminar and to visit those people out there. And I kept putting it off and putting it off and putting it off. And finally it got to be embarrassing. So I thought I better go, because we were really working hard. So I went out, so I flew out to San Francisco, and I gave my seminar. And what I didn't realize the reason that they wanted me to come out there was that they were looking for a chairman of their microbiology and immunology department, and which I surmised after I had been there for a while and talked to these people. And so I gave my seminar, and there was a fellow in the back of the audience that raised his hands as soon as I talked about the IL-2 and the IL-2 receptor and the growth model, or a way to study both normal and malignant cell growth, and I gave some of the data from Julie's MIB experiments. And the thing was that we had, by that time we submitted the paper to Science, and it had been accepted. And one of the reasons that it had been accepted was because this MIB oncogene was thought to cause not cellular proliferation, but to cause cell cycle or cellular discontinuation of cellular proliferation, to stop the cells and to make them to differentiate into whatever they were going to be. And one of the major players in that whole story was a fellow by the name of Mike Bishop. He stood up in the back of the room and he challenged everything that I'd said, number one. Number two, told me that I was just totally wrong about this MIB business, and that it's not facilitating proliferation. Rather, it's involved in cellular differentiation. So I was aware, obviously, of this fellow and their experiments and so forth. And that's why we submitted the paper to Science, because this was a new finding. This is a new phenomenon. It was new for several reasons. One, this is in T cells. And two, it was in the IL-2 model system. And three, here this gene was being expressed in a fashion right building up right before DNA replication started. So, I listened to him, but I didn't say one more, one way or the other. But then after that, then I was, when you visit a university, they take you around and you visit with the faculty for 30 minutes, 45 minutes, each one. And so I visited several people and had nice chats and about what they were doing and how that might have been in relationship to what we were doing. And then I walked into this laboratory with a little office right adjacent, right in the laboratory. It was sort of like a closet, and it was stacked, miles high with reprints of papers and so forth and so on. And the fellow that I was going to be visiting with cleared off the only other chair besides his in the office, and set all the papers aside in another pile that I sat on. And the first thing he said to me, he said, so what did you do in this IL-2 story? We just had Warner Green through here, and he was much, much better than you. More better or whatever. And I was sort of taken aback. I mean, he could think that, but he didn't have to say that to me like that, right? Guy was, whatever. He badgered me throughout the 30-minute or so meeting, and this is when I realized that what they were the reason they had me there was that they were looking for a new chair of their department, and both Bishop and Varmus were in this department, and it was clear they didn't want me to be the new chair. I mean, they'd never met me before, but they probably had somebody else in mind that they wanted to be their new chair and so forth. So that was that. So I went back to Hanover after that, and so that was 1986. Then in 1989, Bishop and Barmas were given the Nobel Prize for their studies from '76 of really proving Howard Kemen's oncogene hypothesis. So, you know, such is the the turn of events in academic scholarship and so forth. But back to Julie's C. paper that came out in Science and subsequent to my visit. As I said, it proved our hypothesis that the first of all, the IL-2 T cell system was really good to be able to study these kinds of things, and we should do it for more oncogenes as they were described by other people in their malignant transformation assay. And it would be a good way to find out, you know, which genes were expressed in IL-2 and T cells. I mean, you could also do it in the other cytokines, because more and more cytokines had been reported. And the thing was is that they were finding, as they got the sequences of the cytokines, the protein sequences and the sequences of the genes, a whole family of molecules ultimately were discovered, and really this discoveries are still ongoing, and we're up to 43 interleukins at this stage of the game. And they have similar sequences, and they have similar receptors. The receptors themselves were being discovered and sequenced, and so a whole, you could see how all of these things were fitting together. And so it was not a tremendous leap of faith to think that, okay, well, whatever is happening biochemically on the inside of the cell, transferring the signal from the receptor into the cytoplasm, and then from the cytoplasm into the nucleus to activate new genes, they were probably similar. So this wasn't just a system for T cells and immunology. This was going to open up the whole genetics of cellular proliferation for everything, for all cells. Because if you think about it a second, if you're a multicellular organism, so that you've got different kinds of cells and different kinds of tissues, you've got nerve tissues and muscle tissues and so forth. Once you become multicellular, in order to not have chaos, you've got to have some way to regulate all this stuff. And that regulation takes place at the cellular level with molecules like interleukin-2, and they communicate with cells by binding to very specific receptors on the cell surface, and then they activate, if they're growth factors, they activate similar kinds of molecules on the inside of the cell. So that was good. And so Julie kept on working on all that. And then one of the genes that she came up with was called Raph, R-A-F, which had just been described in 1983. And we were working in the same time sequence then. So first VRAF from one of the tumor viruses was discovered, and then they looked inside normal cells and they found cellular RAF, just like Howard Kemen had predicted. And it turns out that RAF was well, I forgot to say that MIB was a molecule that traveled into the nucleus and activated genes. So it was what we call as a transcription factor, a factor that helped the message get transcribed from DNA into RNA. Well, RAF, by comparison, was coded for a protein that stayed in the cytoplasm. And it turns out this protein was found to be a catalyst for biochemical reactions. And a catalyst what that means is that it facilitates biochemical reactions, a catalyst does. And the catalyst that this worked on, it caused the transfer of high energy phosphate units from one molecule to another. And that then gave it the name of a kinase. A kinase is an enzyme or a catalyst that transfers high energy phosphates from one molecule to another. Then we also have phosphatases that will take it off, but I won't get into that right now. So, but this was for the first time we started to have an inroad into the black box of what was going on inside the cells when they were started to proliferate. And of course, that's what I started out with in the very beginning as a young man, young, naive individual. I didn't know how long this whole thing was going to take. In order to really get into the nitty gritty of Raph, I felt we were not biochemists and we were not kinase biochemists. And so again, here's where Ellis Reinhartz helped me out. So I called up Ellis and I told him what we were doing and so forth. And I was looking for somebody to help me with these kinase molecules. And he said, well, we've got the world's expert down here at the Dana-Farber. After a while, you get the feeling that all the experts were at the Dana-Farber, but that's not true, really. There were some up at Dartmouth, too. And so anyway, he said, there's a guy down here by the name of Tom Roberts who's really good, and he can help you. So I called up Tom Roberts and told him that Ellis told me about him and so forth, and introduced myself. And so one thing led to another, and I invited myself to come down to Boston and talk to him and explain all the things that we had found and what we were doing. And so we did that. And at the same time, a new graduate student had come into the lab, and her name was Antonina Zhemoitsenos, and that's sort of a strange name, but that she was Lithuanian extraction or descent. And we didn't call her Antonina, we called her Nina, and she was really good. And she had, like Julie Stern, she was a grad student though in biochemistry, and her mentor for her thesis work, I think, left Dartmouth and went off into another institution, and she was looking around for a new lab to come into. So she came into our lab, and she had already learned several things about biochemistry and molecular biology. So she wasn't just like a greenhorn coming into the lab. She had expertise that she brought with her. So I took Nina. Nina and I drove down to Boston, and we met Tom. And Tom, meantime, handpicked one of his postdocs. Harvey Mammon was his name, who is now a radiotherapist. So we decided we'd do a series of experiments, and Nina would do the cell work up at Dartmouth and keep some of the reagents and so forth to do assays, and then we'd send things down to Boston, and Harvey would work on them down in Boston on the more biochemical kinase kinds of experiments. So that worked really well, and wasn't too long before we had really good data indicating that although, and we were interested in, as I said before, we were interested in genes that were activated by the IL-2 receptor, but not by the T-cell receptor. And it turns out that Raph was expressed at the RNA level as a consequence of activating both receptors, but only the IL-2 receptor would cause translation of protein so that you could get Raph protein in the cytoplasm, and also activate the Raph so that it would then have catalytic activity on other phosphorylating activity on other proteins within the cytoplasm. So that was good. So we reported that. The whole thing, the activated catalytic proteins then would gradually increase in concentration over the entire time that the cell was sort of waiting to replicate the DNA. And it would sort of accumulate in that part of the cell, what we call the cell cycle. So we reported that in 1991, and that then really nailed it down for me anyway, that we were right. My hypothesis was right, that whatever was going on in cancer cells was also happening in normal cells. And so that meant we were on the right track, and we should then continue to do these kinds of experiments. Now, one other thing that happened almost simultaneously with all this business was that as a consequence of Doreen's and my 1984 science paper that was all about the IL-2 T cell system as a new growth model, cell growth model, we met another investigator that was a card-carrying cell biologist and expert in this business, and his name was Arthur Pardee. Now, and Art Pardee was, and he was also at the Dana-Farber Cancer Institute in Boston. The thing is, he was a pioneer in the whole area of trying to understand how cells grew in culture and what activated them to grow and so forth and so on. And I went down to give a seminar to his group, and I immediately liked him because he was just as small as I am. So, and I always like little people, you know. so first of all, we look each other in the eye, right? I didn't have to look up to him, and but he was a scientist. I mean, he was serious. When you talk to him about science, he was laser focused on everything you said, and he would come back with questions, very pertinent questions about this and about that, and so forth and so on. But he was very, very soft-spoken and professorial, almost old worldly. He was just a super person. And all of his people in his lab just loved him because he was sort of like the grandfather that you always thought you ought to have to help you get along in life, you know. So in order to see if we could, he was interested in the same thing I was interested in, and that is that what are the molecules that activate the cell to start to grow? And so I just said, well, you know, we have this conference center up at Dartmouth, called the Maneri Center, and you should come up for a meeting. So we organized that, and within, you know, a month or two, and he brought his whole lab up, and I brought my lab, all the people in my lab, and we sat together and then discuss science, you know, nonstop for like 3 days. I think I told you that I'd always be exhausted at the end of these things. But it turns out that Art Pardee was famous before he ever got into all this business. He was famous for some experiments that he had done on sabbatical at the Institut Pasteur in Paris in the 50s. At that time he was an associate professor at UC Berkeley out in California, and he took a sabbatical leave and he went to Paris in the mid-50s, and he went into a lab that was at the Institut Pasteur, and Jacques Monod was the leader of the lab, and one of the other younger persons in his lab was Francois Jacob. And they were very much theoreticians, and they'd come up with the idea that there had to be something inside the cells that was activating the expression of genes. And so when Art went there on sabbatical, he was a biochemist. So he basically did a series of really refined biochemical experiments that ultimately proved their hypothesis as to how genes were regulated. And that was in the 50s. And it was capped off by a famous paper that was published in 1959 that was nicknamed the pajama paper for Pardee, Jacob, and Monod, the pajama paper. And ultimately, Jacob and Monod got the Nobel Prize for their stuff, together with the head of the Institute at Pasteur. And I always thought that they should have given the Nobel Prize to Pardee as well. And I don't know what the politics of that were, but I do know that they only give it to a maximum of 3 people. They can't give it to 4, so whatever. So, and he was a young guy, I guess. They figured he'd do something else really good. So we, at the Maneri meeting, back to that, I'm sorry, I got a little bit sidetracked on that. At the Maneri meeting, Art showed all of his data that it come out. There was a famous paper that he did when he got into the cell proliferation game that was published in 1974. That basically he said that when he tracked cells and they started to go from a quiet stage to replicate their DNA, and in this instance he was dealing with mouse fibroblasts, and the growth stimulus in his system was serum. Now, nobody knew what it was in serum that made them grow, but they knew that he had to add serum to these cultures. And there were ultimately, other people found that there were growth factors in the serum. But there was a point in time that Pardee called the restriction point, when you needed the serum there. But after that restriction point, if you took the serum away, the cells still proliferated. So that was a good sort of a marker for the kinds of studies that were being done by all kinds of people in the business. And for the most part, they were using the same system that he did, which were the fibroblasts in the serum as the growth stimulus. And of course, our paper that came out 10 years later in '84 replaced this mysterious serum growth factors, whatever was in there. And it could have been a bajillion of them. They replaced it with one molecule, and that was IL-2, and which only had one specific receptor that we could measure and so forth. And so that we could finally explain all of Pardee's experiments by that '84 paper. And also extrapolate from T cells and IL-2 and that sort of thing to all cells. And when we published our paper in Science in '84, I went through the whole history of the experiments and the thoughts and the hypotheses that had been generated in terms of why the cells grew the way they did. And basically, the experiments go back to 1932 and first done in bacteria, and then they did it in yeast, and then they did it in all cells known to man and God. So essentially, our IL-2 T cell experiments had cracked open, wide open, the whole thing in terms of all the unknown variables that had been talked about and people had done experiments on. And so Art Pardee and I were right on the same page. And he had devised a method to identify, isolate, and clone genes from cells that were in the act of trying to get ready to grow. And it allowed one to separate out all the expressed genes that were expressed as a consequence of the being induced to grow, say with IL-2, from the so-called housekeeping genes that were there all the time in the cell and so forth. So that we could then purify with his method the induced genes by greater than 90%. And so we could just throw away all the housekeeping stuff that was going on, and we could focus down on the induced genes. And he described that at our Minari meeting and so forth. And it was just about that time that another new person came into the lab. It was almost like, it's a revolving door as you come to my lab, whatever, and have some interesting experiments to do. And this person turns out that she became crucial for like the next decade of experiments. And her name was Carol Beadling, and she was a graduate student at Dartmouth. And she has an interesting background. She went to Reed College in Portland, Oregon. And if you know anything about Reed College is a very, very liberal, politically liberal institution. These are the granola people out there with their backpacks and that sort of stuff. And she once told me after she came to Dartmouth. And so, but she started in her undergrad years, she started working on IL-2 from frogs. So she worked on frog IL-2. Now, I don't know, she was in a very basic fundamental biology department. And so you work on things like frogs. And as a consequence of that, she read all of our papers and so forth. And so she came to Dartmouth to interview with me. And I'll never forget it. At that time, in the wintertime, the snow would be this high. And I used to walk to work through these big snow drifts and so forth. And my favorite footwear to get through all the snow and the slush and so forth and so on were cowboy boots. I just liked the cowboy boots. She told me later on, so she was interviewing with me in my office, and she told me later on that clinched it. My cowboy boots clinched it. So anyway, after she'd been to Dartmouth for a while, and she found out Dartmouth's a very conservative institution, politically speaking, as I went to when we talked about Lynn's restaurant and so forth. So she said she felt like she'd landed on Mars after all of the liberal types in Portland, and so forth. Anyway, she decided to work up Art's gene selection assay and get it going in our laboratory, and she did. And she was a very thorough, meticulous, careful, tenacious type in the lab. I mean, once she got involved, she just didn't let go. And because it was very labor intensive work to do these kinds of experiments, and also much longer term, as I think I may have said in one of the other things. And so it took a while to get, you weren't having a eureka day every other day with this stuff. Ultimately, with the help of one of the postdocs in the lab, Kirk Johnson, after 2 or 3 years, she was successful in working up the whole system, and she'd identified 8 new genes, that only one of which had been previously known, and only 2 of which were also activated by the T-cell receptor. So there were 6 new genes that we had to work on, and that was great. We called them cytokine response genes. That never took on. Every time I tried to coin a term like T-cell growth factor, people wanted to change it to something else. Same thing with CR genes, cytokine response genes. But we published her data in a paper that came out in the Proceedings of the National Academy of Sciences in 1993. That then served as the basis, essentially, for what we started to work on next, because we had to characterize all these genes, find out what they did, because it wasn't as simple as working on the oncogenes that had already been described by other people, and they'd gone down all the grunt work to find out, so what are they and what do they do, and so forth and so on. So we had to do all that work now too. That brings the 1980s to a close and at Dartmouth. Almost. There's still another chapter to go here in the quest for new knowledge, and which will be chapter 8, and I'll talk about that next. But I'm going to stop there. Thanks. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.